which is 2n, the diploid condition has been restored during fusion of gametes. Diploid state is restored. So now we can simply say that the genotypes are the same. So offspring phenotypes, that is the offspring physical outlook. These individuals of fruit flies are all long-winged, flies. So that is the first case of the test cross. When you are writing the genotypes of the offspring, you can as well write test cross genotypes if you don't write offspring genotype. Or you can write test cross phenotypes instead of offspring phenotypes. It is still fine. So now, another case is if the fruit fly with long wings is heterozygous, then what will happen, or what will be the genotypes of the offspring? Remember that the other parent will still be maintained. We shall still cross with a vestigial winged Drosophila. So I will write here, that is the first case. Second case here is if the long-winged Drosophila is heterozygous, then so we shall have offspring, we shall have parental phenotypes. Both parents are long-winged. No, they are not long-winged. One of them is long-winged but heterozygous. Let's say long-winged Drosophila. Long-winged Drosophila. We are crossing it with vestigial winged Drosophila. Vestigial winged Drosophila. Then we shall have parental genotypes. Parental genotypes, which are a diploid, that is 2n. This one is heterozygous. We shall have L with capital L with small l. We are crossing it with L small. The genes segregate to form capital L, then small l. Also, this gene here will segregate to form small l and then small l. Those are the alleles upon segregation. These ones are carried in gametes which are haploid in nature, and the segregation, of course, occurs during meiosis. Meiosis occurs and genes segregate. Now, at fertilization, what shall we observe here? Then we can make our crosses. We can cross. We can make genetic crosses as shown there. We can make genetic crosses. We can make genetic crosses. Think, you can observe my crosses here. I have made them. Now this cross here will give us a genotype with a dominant gene and a recessive gene. This one also will give us capital L and small l. This genotype will bear recessive alleles. So small l, small l. Also this one here, small l, as shown there. So that is offspring genotype. Offspring genotype, that is 2n. Of course, here we have random fertilization. Random fertilization is there. Now, what do we observe here? The offspring phenotype. Offspring, this is genotype. Then offspring phenotype or phenotypes. We can see that two Drosophila are long-winged. Two long-winged. Then the other two vestigial winged. Therefore, in conclusion, we can simply say that when we are carrying out this testing cross here, what we shall observe is that in the first case, if all the offspring or individuals are long-winged Drosophila or fruit flies, then we can deduce that the parent or the Drosophila with the dominant character, which is long wings, is homozygous for long wings because the gene for long wings has suppressed the expression of the recessive allele, which is the allele for vestigial wings, as shown here. Now, in the second case, we can observe that suppose the individual or the Drosophila with long wings is heterozygous, as shown here, then we can see that the phenotypes of the offspring or the progeny will be two. Some Drosophila or some fruit flies will bear long wings. Actually, two of them will bear long wings, and the other two will bear vestigial or short wings. And therefore, this gives us a phenotypic ratio of 1:1. One long-winged Drosophila to one vestigial winged Drosophila. And therefore, here we can deduce that the organism or the Drosophila with the dominant character was heterozygous. And so, in this way, you will determine whether the Drosophila or the organism with the dominant character is homozygous or is heterozygous. So friends, that is the testing cross involving monohybrid inheritance. I wish to stop here, so you can join me once again when I appear So that is it. So you should be able to recall that. Thank you.